Hello everyone, we are back with another video. Today we are going to discuss fructose, galactose, sorbitol, and ethanol metabolism. Firstly, we are going to have a look on fructose metabolism. Its main dietary source is the disaccharide, and that is sucrose. Gut sucrase hydrolyzes sucrose to component monosaccharides glucose and fructose. Fructose is a hexose, means sugar having 6 carbons in its structure. Absorption of fructose into hepatocytes or liver cells is through the Glyceraldehyde transporter. Fructose is phosphorylated by hepatocyte fructokinase, forming fructose 1-phosphate. As you can see, phosphorylation at position 1 of fructose. This is further metabolized in the presence of aldolase B into dihydroxyacetone phosphate. First 3 carbons of fructose breaks from next 3 carbons. OH group from carbon number 2 is removed. So dihydroxyacetone phosphate forms, and in case of glyceraldehyde, carbon 4, 5, and 6 separates from first 3 carbons. At position 4, 1 hydrogen is removed from OH as cyclic structure breaks, and OH at position 5 is added. So glyceraldehyde is formed. In the presence of enzyme called as triose kinase, glyceraldehyde converts to glyceraldehyde 3-phosphate, which enters glycolysis or gluconeogenesis according to cellular energy status. Now moving toward galactose metabolism. The main dietary source is the disaccharide lactose found in milk products. Gut lactase hydrolyzes lactose to its components such as glucose and galactose. Like glucose, diffusion into the bloodstream from the enterocytes is through the GLUT2 transporter. Galactose is converted to galactose 1-phosphate by galactokinase and phosphorylation. Now galactose 1-phosphate reacts with UDP glucose, forming glucose 1-phosphate and UDP galactose. This is catalyzed by galactose 1-phosphate uridyl transferase. Glucose 1-phosphate is then converted to glucose 6-phosphate by enzyme called as phosphoglucomutase. The UDP galactose is reconverted to UDP glucose by galactose 6-phosphate epimerase. Thirdly, we are going to discuss metabolism of sorbitol. It is a sugar alcohol. It can also be obtained from the diet. Sorbitol produces endogenously from excess glucose through the polyol pathway. This is glucose having cyclic configuration. It is converted to linear configuration in presence of an enzyme aldose reductase. The polyol pathway utilizes NADPH and H+ as redox partner. Nicotinamide adenine dinucleotide phosphate and sorbitol is generated. As you can see, reduction has occurred, and hydrogen is added to carbon of position 1 and another hydrogen added to oxygen attached to position 1 carbon in structure of glucose to form sorbitol. In next step, in presence of enzyme sorbitol dehydrogenase. This reaction also utilizes nicotinamide adenine dinucleotide as a redox partner, thus generating NADH and H+. Now from the structure of sorbitol, 2 hydrogens are removed, 1 hydrogen attached to position 2 carbon, and second hydrogen from OH group attached to carbon number 2, and now double bond with oxygen is formed to fill 4 valencies of position 2 carbon. It is then converted to fructose, which is in linear configuration, and then this fructose converts to cyclic configuration. The fructose may ultimately feed into glycolysis, or the pentose phosphate pathway, or gluconeogenesis in the liver. Lastly, we are going to discuss metabolism of ethanol. Do you know what ethanol is? Well, ethanol is a component of alcoholic beverages. It binds to many CNS receptors, including acetylcholine, serotonin, and glutamate, with an overall depressant effect on neuronal activity. Let's discuss its metabolism. This is ethanol, all mean alcohol. Nicotinamide adenine dinucleotide is oxidized form. It is utilized and converts to NADH and H+ in the presence of enzyme called alcohol dehydrogenase. Ethanol is formed by removing one hydrogen from oxygen and other hydrogen from carbon. This is aldehyde, indicated by al at the end. Again, nicotinamide adenine dinucleotide is utilized, and NADH and H+ are generated, and ethanol converts to acetate in presence of enzyme called aldehyde dehydrogenase. As you can see, oxygen is added in place of hydrogen. The acetate produced can then be coupled with coenzyme A by the enzyme acetyl coenzyme A synthetase, forming acetyl coenzyme A. That was all about today's video. Stay tuned to sqadia.com for more precise and knowledge stuff videos.